Good afternoon, all. Can I just check that you can hear me? I notice you don't have headphones on. It's not needed here, is it? It's quiet enough that you can hear what we're saying, which is great news. Welcome. Thank you for coming to this session. We're going to unpack epigenetics with some experts that I would like to introduce in a second. Prior to doing that, I'll just introduce myself. My name is Paul Jones. I run the Centre of Excellence for Omics in Abu Dhabi, part of M42, which has been working with the Department of Health on one of the largest genome sequencing programs anywhere in the world. We've got in excess of 600,000 genomes, actually across the local Emirati population. A certain proportion of those actually have got 100,000 of those, in fact. We do have long-read technology that allows us to get an insight into the epigenetic profile of 100,000 samples, which I think is possibly one of the largest cohorts in the world. So that in itself is interesting, and we will come back to that at the end of the discussion. If I could just ask my guests, please, to introduce themselves. Let's keep it brief, because we are going to come back and revisit the past, the present, and the future in terms of the discussion. But if I could start with you, Gabby, at the end, would you mind just giving us a brief introduction of where you come from and the relationship to epigenetics in terms of your background?
Right. Okay. Hello, all. Nice to see you. So I'm Gabby Ficz. I'm a researcher and academic at Barts Cancer Institute in London at the Queen Mary University. And I think I've been in the epigenetics field for the past 20 years. And we've been working on epigenetic editing for the past 10 years. So.
Super. Thank you very much. Laura, over to you. Brief introduction, please.
Salam alaikum. Hello, everyone. My name is Laura Bierut. I'm from Washington University in St. Louis in the United States. And I work on the genetics of behavior and substances that we use, our behaviors, obesity, and kind of lifestyle. And these our behavior is in part genetically driven, and so trying to understand the genetic predispositions that we have.
Super. Thank you, Laura. And JJ, can I ask you, please, to give us a brief introduction?
Yes. I'm JJ Liu from Singapore, and I'm currently distinguished principal scientist at the Genome Institute of Singapore. I have been studying the genetics of a complex disease for over 2 decades. Currently, I'm also deeply involved in the Singapore's National Precision Medicine Program, where I'm in charge and leading, the population sequencing projects. Very happy to be here.
Are we having some problems with the mics? You're not able to hear us? My mic, I think, is okay.
Yeah.
JJ, maybe for you.
Well, I'll have to use this one.
Yeah.
Okay. Oh, okay, fine. Yeah.
Did you hear what JJ said? Do we need more introduction there? No, all good, all good.
Okay.
Right, let's kick off the discussion. Gabby, can I come to you first? Would you give us a historical perspective on why epigenetics is relevant and your perspective from your area of expertise, where the field has been over the last few years?
Okay, well, let me start with the definition first, because many understand different things under epigenetics. So before I give a technical definition, I want you to think of our body as a computer, and a computer has a hardware and a software, and the hardware is the genetic sequence, and the epigenome information is the software. So the idea is that this program can control our genes, what to do, when to do, under which condition, and it influences us from our early development, from a zygote, from when we were a one-cell embryo, up to development to become a human being and aging, and it can also impact the next generation. So there are many nuances to epigenetics. Now, historically, obviously DNA has been described in the 1950s, even before that, but at that time, people just thought about the genetic information. But people have noticed some chemical tags on the DNA and on the proteins that protect the DNA. So the DNA is not naked in cells, it's protected by proteins. And those proteins and the DNA can have tiny chemical tags. And those chemical tags are diverse and can be a code to say something, that this gene should work now, or later, or in this cell or that cell. So we have one genome, all of us, a unique genome, but we have about 200 epigenomes in our body. Each cell type has its unique epigenome.
Super. And is that, does the epigenome, as it were, influence the turning on and off of those genes? Is that the message here? Is that how it works? We should think about how the genes are expressed based on epigenetics.
That's right. So the technical definition is that epigenetics is changes in gene expression that are not dependent on the underlying changes in DNA sequence. So it's not based on changes in DNA sequence.
Thank you, Gabby, for that definition. Laura, can I turn to you in terms of how that definition has been relevant to your field over the past years?
So I work in the genetics of behavior and substance use and mental illness. And what we're seeing now with epigenetics is that our environment and our behavior influences what genes are being turned on and off. The classic example that we could think about is cigarette smoking. So cigarette smoking leaves a long-lasting epigenetic signature. Some genes are turned on, others are turned off. And even if you quit smoking, decades later, we could see these epigenetic changes. So the way that I think of epigenetics is how our environment is influencing what genes are being turned on and off.
Super. Thank you, Laura. And is there in terms of your area of expertise, how has that influenced where historically we've been using that technology? Could you just give us some insight into your...
So I think of epigenetics in two different ways. There's the prevention, and then there's treatment. So I sit heavily as a physician scientist in the prevention area of how can we prevent some of these pathologic epigenetic changes from occurring. So that's why we focus so much on healthy lifestyles. But there's the other point, which is once some of these pathologic changes occur, can we do something to turn them back off? And some of them turn back off over time, naturally, others do not. And so we really need to be focusing on those that are not turning off to promote health.
Very good. Thank you very much for that. JJ, can I turn to you? Would you give us a historical perspective in your field as to where this has been relevant?
Yes, and I am a human geneticist. I used to study the variation of DNA molecule. You have heard a term that called the genetic risk, because for human health, 30% of our risk for disease is controlled genetically, determined by the variation of our DNA molecule. And I historically think that is the ground truth. Everybody have the same risk from the day one you were born and until the day you die. That never changed because our DNA molecule never changed. But now I more and more believe that's not true because, like Gabriella said, the gene need to be expressed in order to have effect. The particular risk allele we carry need to express, increase our risk. That telling us the risk, even though they carry same genes for your whole life, but the risk at any particular moment, any particular environment, and with any particular lifestyle, the risks are different because they all influence how particular genetic risk allele express itself. So that's why I think there is a great application for us to really understand how the genetic risk express itself, how they influence our health.
And JJ, I know you've been involved with the Singapore initiative.
Yes.
Would you give us a bit of insight into that and how epigenetics plays into that sphere?
Yes, and National Singapore as UAE, we have a national precision medicine program. And that program, like Laura highlight, there's two perspectives. One is really see how we give the right drug, right time to the right patient. That is really talk about precision treatment. But there's another big objective for the program is called predictive and preventative medicine. Really trying to assess how the individual have a different risk for certain conditions. Can we actually, if we know that information, can we use that information to designing we call the precision or precise or individualized prevention intervention programs. That is actually another major objective of the program, because only work on the treatment is not going to reduce the disease burden. It's not going to control our healthcare expenditure. Only we turn around, reduce the overall disease burden, then we'll see the true fruit of this precision medicine program. The huge investment, but all the different countries right now are working on that.
We'll come back to that, JJ, because I'd really like to understand where the program's at today in terms of unlocking the value around both treating disease and looking at this prevention agenda. But let me please pass back to Gabby, if I can, at this stage. Gabby, could you just bring us up to date? Where is your field today? What's happening today? What research are you involved with that's driving the agenda around this space?
So one of the biggest challenges with epigenetics is that there are many changes that take place at the same time. So when something happens, a disease develops, many changes occur. We don't know which ones are pathogenic, which ones are not. With epigenetic editing, for the first time, we can address that very question. So this is what we did since CRISPR tools, which is the molecular scissors, many of you know, came about in 2012, I believe.
Just explain it, just
all
in
right,
case
so
everyone isn't
CRISPR, the
familiar.
CRISPR tools, so these are molecular scissors. Basically, there are some tools that we use in the lab where we can direct a molecule to a specific region in the genome to cut, right? And that means that we can make a mutation there, we can have an insertion, so insert some foreign DNA in there, we can do all kinds of genetic manipulations. Now, some people had a thought, including me, I mean, good ideas, happen in many places in the world, where you can actually inactivate the catalytic activity of this molecular scissor, in a sense that it's not going to cut the DNA. You can fuse it to another protein that is an epigenetic editor, and then it will drag the editor to a specific location, and I can just modify epigenetic signatures. So now we ask the question, some of these signatures that we see in cancer, are they pathogenic? And in fact, what we observed is that when we were playing with human cells in the dish, just by changing a specific chemical tag in the DNA, we were able to insert a false memory in that cell and force it to do something that they would not normally do. So that was in the dish, we could increase proliferation. Then, when we put these cells into an animal, in the bone marrow, that cell, no genetic sequence was changed. That cell modified the immune system, and it created a potentially inflammatory environment. So with that, we could see that the memory was permanent, and it also impacted the physiology. And that's just one locus. Imagine what we can do with many such questions. So we can be precisely asking questions about epigenetic impact.
And is that yet having an impact on humans?
Good question. So so there are several of us in this field now, and there are already experiments that are done in primates where we can interfere with cholesterol homeostasis, so we can modify genes to control the level of cholesterol, and that is working. So that's pretty much where we are now, and then I guess the next step is clinical trials. But there are so many questions that we still need to address, and I'm very passionate about this idea of prevention, cancer prevention.
Super. Well, thank you for that background. That's very interesting. Laura, could I ask you to give us a perspective on where you're at today with your research and where you see the field as it is today?
So I work in behavior, so I really am interested in the brain. And what we could see in the brain is we could see certain epigenetic signatures associated with different types of behavior. So with substance use, with mental illness, cigarette smoking, we could see these changes, again, that are long-lasting in different parts of the brain. Most people don't like donating any brain samples, so we are challenged with how do we really move this forward using more peripheral samples, like blood, and understanding these signatures in blood and relating them to the brain. But where we are right now, which I'm really excited about, is again this prediction of who is at risk. So we are on the cusp. We should be returning genetic information now to people so that they understand their genetic risk. And this is true for breast cancer, this is true for obesity, this is true for diabetes. All of these diseases, we should be returning that information now to individuals so that they can be empowered to take environmental changes to reduce their risk of disease. And this is considered controversial at this point of how will people respond? Is this dangerous? Do we think about the bad things that could happen? But in all of my research, people want the information. It's all about me. Everyone says, it's all about me. I want to know my risk. And it gives an individual that power. And we do not see individuals going out and doing terrible things. So saying, oh, I'm at low risk for obesity, give me donuts, and I want to eat as many donuts as possible. In general, it's improving behavior.
Laura, can I just challenge that for a second? Because my wife is a physician, and she's interested in everything that we're doing with the genetics and the epigenetics here. But she says, well, at the end of the day, when you give advice to individuals, at the end, it always comes down to eat more healthily, sleep well, have a good exercise well. What is it that people can do with that information that they couldn't do anyway without that information in terms of living a healthy lifestyle? How do you weave that into the kind of advice and the responses that you may have?
So, as we think about these things of, eat well, sleep, exercise, some people are really particularly at risk and others are not. So what we've been doing is giving the whole population this whole thing of you should eat well and you should exercise and you should, sleep well, do all these things. But we really need to tell someone, you are at particular risk. Again, everyone should do this, but you are at particular risk for diabetes. So let's really make sure that we keep your weight down. Right. And so understanding an individual's own risk, I think, is important. It gives them agency. It gives them the feeling also that they're not as much to blame, that they have these different predispositions. So again, I always hear, well, people, know these things already, because again, I am a physician also. Yeah. But this really seems to be motivating behavior much more strongly than just the general risk that we tell people.
Got you. So, one of the things that I'm fascinated by is making an individual impact, but potentially being able to do that at a population level. So if I heard you correctly, what you're suggesting is at a population level, you can identify groups, cohorts of individuals who might be at super high levels of risk.
Yes.
And you should be working with them proactively as individuals to make sure they manage that, they're aware of that risk, manage it appropriately, recognizing actually there may be other parts of the population, segments of the population that actually where the focus is not as critical in terms of the impact it may have at a population level. Have I understood that correct?
And let me give you a specific example with breast cancer. So in the United States, we have these general recommendations for mammography screening for breast cancer. Some women probably don't need to get screened for breast cancer until they're 55 or 60. And other women need to be screened when they're 35. But in the United States, we have this general recommendation, start screening at age 40, because we're looking at the general population risk. But what we need to do is do this precision prevention, where some women need to be screened earlier, others could be screened later. Colon cancer is another example that we have. And so, again, it's identifying the high-risk populations, intervening more strongly here, and reserving the resources from the other parts of the population that don't need that.
So I've heard that described as precision public health. Would that be an appropriate term that you could live with?
I could live with that one, and I could also live with precision prevention.
Right.
So I live with precision prevention, but precision public health is in that same area.
Great stuff. JJ, can I turn to you? Would you bring us up to date with what's happening in your field and the research specifically that you're focused on at the moment?
Yeah, I'm involved in national precision medicine programs. My feeling is it's no longer the question for us to get the sequencing out, get the variation out, get the risk information out. But how can we influence people to take action? You give the information, end of the day, it depends on that person take initiative to change. Otherwise, who bother? All the information going to be in vain. What is it? So I think we do need to work with the people to find a way how the information can be communicated so that that individual receive that information can be motivated to act, to change. Otherwise, you're not going to see the fruit of this national precision medicine. You're not going to see the fruit of the preventative medicine because prevention, end of the day, rely on individuals act. That's where, for all of this, how do we do that?
And how do you do that, JJ? Are you learning some lessons?
I think, we need a more behavior science, understand how people react to, because some people driven by fear, some people driven by explorations, some people just never do anything until somebody else do it for them. I would say there's different way to find communication, the information, in the different personalized way that will motivate them. And if for someone really don't want to do anything, well, there's always possibility one day we can give a pill, right? Always people think about it. Is there really future rely on individual behavior change? Is there any way we can use the other way to influence the risk? Because if the risk is determined by how the gene carry the virus, how the gene expressed, if we can changing all this, theoretically, we can intervene. Is that what in the table in the future? To modify how the genes express itself, how to use in other ways, pills or other way to change it. This is something we need to think about it.
Laura, I can see you going to jump in. Please do.
I want to respond to this. So I am in complete agreement with what you're saying. And what I've loved about this forum is how it's bringing people together that think in different ways and breaking down our silos, because we tend to be experts in these different areas. But there is a whole field of behavioral science and implementation science. And the idea behind that is, how do we drive behavioral change? How do we use the system to do that? And do you know that behavior is kind of infective? You know that when one person starts a behavior, it kind of moves in groups. So we could think of behavior as kind of like an infection in the population, both good and bad. And so we need to be thinking of how do we return this information? How do we return it in a way that's scalable? So historically, we have done genetic counseling. There are not enough genetic counselors in the world to treat the billions of individuals that we have. What we need to do is we need to use technology. We need to use web-based technology to give information back, to know how people are responding to it, to know the positive ways and the negative ways that they're responding. So this is really exciting because we have these technologies to now scale at a population level. So we need to think, again, as a physician, I've thought about sitting with a patient, talking with a patient, but I do not have enough reach. And how could I extend my reach at a population level? And that is really exciting.
I'm with you. I mean, one of the things I've been involved with, particularly over the last decade, is often working with governments looking at population level agendas. And they may identify these cohorts of high-risk individuals, and they may even have some ideas about how you might start to influence them, but they realize actually the implications in terms of the size and scale of the challenge and the support that's required when you don't have enough counselors of the right skill set within a country. Could you just unpack for me some of the risks that you see around some of the areas that we're talking about here in terms of the implications? Let's start to think about the future here and looking at it at a national level. Often these programs can be addressed from a national perspective. What things should we be aware of and conscious of as you start to be able to identify high-risk cohorts and the implications of what that might mean? Laura, do you want to just jump in with that?
Sure, I'll start with this. So I'm just going to give my beliefs here of where we are, seeing the future. So we often talk about the risks. We need to also talk about the benefits, because focusing on the risks is really impeding the science. that we have. And there's all this fear of behavior. And again, in all of my research, I am not seeing kind of any of the adverse consequences that we fear. So what do we fear? We fear stigma.
Yep.
We fear the idea of, oh, you know, you're predisposed to mental illness. So I'm a physician who deals with mental illness. You know, it's a tragedy that we have. And so there's the stigma of mental illness. There's the stigma associated with obesity. And, you know, how do we kind of remove that type of stigma is something that we need to think about. The other thing, which is there's this fear of genes are destiny. And genes are not destiny. Genes give you a predisposition, but we have the power to change our environments. And so there is this fear that, oh, I'm at risk for this disease, so what does it matter? I'm just going to go out and, you know, live a terrible lifestyle. And again, I am not seeing that. I am seeing actually the returning of these results as empowering people to live a healthier life.
Very good. Gabby, can I ask you to just comment on the risks in your field? What are some of the potential things that we need to be aware of looking into the future?
So in terms of cancer prevention, I do have this vision that in the future we will be able to prevent cancer in the population by intervention without having to rely on behavioral changes, in a sense that right now there's a lot of research going on in trying to understand the transition from normal to cancer. Mm-hmm. We know that all of us are clones of cells mutated in our bodies. However, most of us will not develop tumors in most of our tissues, right? So there is something that stops it. Now, if we identify that mechanism of transitioning and stopping, and I think the key is in the immune system, we will be able to develop vaccines to prevent that transition, to intervene the earliest possible way. Now, there are ethical implications in that, obviously. How early do you intervene? And some of these epigenetic editing technologies do not have an ethical framework. We don't know how much we can do, how much we should do. And now that we know that the memory persists, do we need to think about safety measures to reverse it, you know, when there is a dangerous intervention? So there are so many questions that are still outstanding. However, this idea that in the future we will be able to develop vaccines, I think it's real, and we can already act now.
Do you have a concern? I've heard it expressed by some people that full body MRIs for everyone will identify things that actually wouldn't necessarily have turned into problems in the future, and so you can overdiagnose issues.
Right,
How do you think we cope with that challenge?
So this is an issue because there is knowledge that if we did autopsies on 100 people who died in accidents, about 7 of them have a tumor in them that doesn't manifest, right? So that's a large number, right? What do we do about that? Because it's similar to the breast cancer prevention. We over-treat women with chemotherapy, that they don't need it, right? And that disrupts their lives. However, this transition from normal to cancer seems to be plastic, seems to be happening all the time. It's just understanding better what's going on there.
Right.
And now that we know that we can also create designer immune systems, maybe we can play with that and develop interventions that are still in the future, that is just in the realm of imagination today.
Thank you, Gabby. JJ, can I ask you to comment on some of the risks that you're seeing in the field that you're working in at the moment?
Yeah, I feel like, you know, I guess not in the future, each one of us will have our so-called report card, which is called the genetic risk for not all disease, but most commonly disease on the, you know, genders, and all of us, like England's, you know, UE's, you know, Genome, you know, precision medicine program in Singapore, they will have this. The information will starting to be returned back into video, right? But in terms of to turn that information into the benefit, I think it rely on both individual and probably also the government. If you talk about smoking-related risk, yes, you can rely on individual to change their smoking behavior, but along the way, strong government regulation always required. To create an environment for people to have a motivation quitting smoking is important. If you purely rely on the voluntary motivations, I would say we're not going to get today such reduction of smoking rate. It's always both sides. I think government need to step in to see how we use the risk information to structure the social program that could drive in certain behavior. That one, I think government need to step in to understand.
Got you. Can I change tack slightly? I'm going to ask some advice if I can. So I've recently come to the UAE. 6 months ago, I joined. I was fortunate, privileged enough to be sitting on top of this Emirati genome program, where we're striving towards delivering 1 million genomes in the next few months. There's already 600,000 plus whole genomes been sequenced. We've got 100,000 long reads with an epigenetic profile on 80,000 samples. What would be your advice to me as I come into this role about what we should do with that data set, how we should think about it, and how it might be relevant to your field of expertise? I'd welcome your perspective. And if I can get that advice, what I'd love to then do is just move in the last 10 minutes of the discussion to look long term into the future, the next 50, 100 years. What do you see is going to happen in this field? And I'll just set that up so there's a couple of key points there. And if there are any questions from the audience, please, we'll come back to that in the last 5 minutes and just make sure that we've addressed those in the last 5 minutes of the session. So please, can I get your advice on what you would recommend we do with this large-scale initiative, population, government-sponsored, but with a large genetic perspective? Please, Gabby, why not start?
So I would already start collecting blood from a large fraction of the population, so we have a database, and we have good annotation for future AI applications, which is the important bit. So
that's the first time today that AI has been mentioned. I'm interested by that. I always thought it might come in earlier, so please, I don't want to stop you, but yes, so you see the AI potential.
There is definitely AI potential. We do need more data for that, a lot of data, right? And we do need data from healthy people who in 20 years will have developed disease. So we can look at the blood today, and in 20 years, we can use the AI tools to see why they developed.
What markers were there.
Yeah,
Gotcha.
and that can be blood, that can be other materials, but from healthy people. So that I can see already as something that we can do today to increase that cohort.
Effectively what we have, which I think is quite unique in the UAE, is 7/10 of the local population sequenced.
Yeah.
Clearly, the intention is that those are healthy individuals, but not all of them will be, and you will also pick up disease. So, love the idea that actually, from a longitudinal perspective, you can use that insight, even for people who appear healthy today.
That's right.
If they get ill in the future, you can look back at that sample and start to unpack what may be in there. So that's an opportunity that you would advise us to capitalize on. And that means collecting samples properly that can be stored into the future.
Exactly.
Brilliant.
And quite a bit of samples, so not just 10 ml of blood. It needs a little bit more in order to be able to
How much more? What will it need? What kind of thing will it need?
I would say more than 10.
Okay.
Now,
11. Got you.
There are already such cohorts, but they are small, where they were collected 20 years ago, and now we can use that database in order to see how their epigenome behaved then. And now we know that they have leukemia, for example, and we can see changes that happened already a long time ago. So there is a huge benefit of that, but we need a lot of data. We couldn't have sufficient statistical power with a cohort of 20 AML patients. So we do need large numbers, and that's just AML, like leukemia. We do need for many other diseases.
One of the things that I've been intrigued by looking at the data set is this idea that there is a very strong family structure within the Emirates, large families, and the potential to look at those families as a very rich source of insight. Could you just comment on that? Does that, I mean, I'm not aware of something of the size and scale in terms of the whole of the population being sequenced. That presents, I think, some very interesting opportunities in the future. Would you agree?
Yeah, I would. So in terms of numbers, I don't know what would...
Some families of like 40, 50 people where you can see the grandparents, parents, children all in the line, some with certain disease that you can see being passed down the genetic line. So that provides an incredibly powerful source of insight in terms of being able to understand what's going on there from a disease mechanism perspective.
Correct, yeah, both on the genetic side and on change...
The family structure.
Yeah.
Laura, can I ask you to comment? What advice would you give us at M42 in terms of what to do with this asset?
So I'm interested in the brain, and I would say that this is a data set that needs neuroimaging.
Right.
And having neuroimaging on such a large population would be an incredible resource so that we know how the brain is changing over time, over different ages. And there are these new techniques with imaging where we could determine brain age. So it's this idea of, we have a biologic age and we have a chronological age. And these two things can differ. And we can now identify people's brain age. Some are 10 years younger than they are, and others are 10 years older. And how do we understand the genetic predispositions to this environmental predispositions to the different brain age? Because what is clear is the children today are going to live to 100. Mm-hmm. And so if the children today are living to 100, how do we keep them healthy as long as possible and their brain as healthy as long as possible? Because our brain does age over time, and what we want is this brain intact.
Super. Thank you for that, Laura. JJ, what would be your advice?
I think genetic information need to be combined with all other information. Your EMR, electronic medical record, the lifestyle information, the environmental information. Only by integrate this information with the other information, they will start to be able to understand what the real meaning of that genetic information. That one is basically probably everyone is recognized already. Epigenetic information, we talk about the long-read sequence you can read directly from how the DNA molecule are modified. I think that is probably still too early to say they can be very useful. Why I see it that way? Because just mentioned, each one have a single genome, but over 200 epigenomes. The one we got now from the blood is probably a few of this program. The cell type presented in the blood, that's all. There are a lot of other epigenetic program, we have no much information at all. We are not there yet. If you think really to use epigenetic information to do something, I think they are way far behind the genomic information aspect. I think we are probably need to work on that one, but we definitely need to invest a lot more on the epigenetic program. But we need to, we call it multi-tissue, systematic, profiling, blood in terms of methylation information or epigenetic information, my personal opinion will be very limited, I would say, at this stage.
Thank you, JJ. I'm going to ask you to just jump straight on from that thought. If you could just look out 50 years, where's this field going? Where do you see this? Because it extends nicely from what you were doing there, and I'll ask the same, and then we'll open the floor to questions.
History tells us when the human knows something, they will always try to change it. Okay? Along the way, there will come a day we'll start to change the DNA sequence. We're also going to change the program, all these genes, how to express self. This is what happen, either proactively or just by certain private sectors, they will go in that path. I do think we need to think very carefully, how are we going to deal with that? Because there is a huge potential and same time, huge risk. And I would say that's something I think we need to really watch out.
Thank you, JJ. Laura, over to you. 50-year perspective, even longer if you want.
Okay, so I'm going to start with 10 years. I think in 10 years, we're going to have genetic data on every child that's born. And so that we understand what their risk is for childhood diseases. and that we could start treatments for them immediately when they get some type of illness, asthma, infections, understanding where they'll have bad side effects, because we all know that when we take medications, we don't respond the same way to medications, and we respond well to some and have side effects with others. So that's the 10-year vision. In 50 years, I think we are going to have so many biomarkers that are available, that are going to be, with a simple blood test that will understand our predisposition to cancer. We're going to be catching it early. We're going to understand immune diseases and where they are, and we're going to be intervening specifically at individuals at earlier stages. Gotcha. And we're going to prevent disease. We want to live a long and healthy life, not just a long life.
Super. Thank you, Laura. And Gabby, can we close with yourself?
So I'm assuming in 50 years humanity will survive, so having that assumption, I do think that we will be having personalized intervention. We will have a huge knowledge about our risks early in life and augment ourselves. And definitely for the next 10 years, what Dubai can do with the leadership, with the resources, with the infrastructure, is to help this movement to make the most of the cancer prevention, of diabetes prevention, and have a designer immune system.
So there's a very strong preventative theme.
That's right, yes.
In what you're seeing in the future. Super. I know there are a few hands that went up. Wow, we got lots of questions. Maybe we should, please, let me just start on the front row here with the gentleman with the glasses. Would you mind sharing a mic? That was a huge number of questions. That's great to see. Maybe we have to extend this. I'm not sure what the permission is to extend.
Well, maybe a question is big, but I want to make a bridge with the previous session. Where I'm Tom Myers, I'm from Belgium, I'm an osteopath. Futurize yourself is my thing, and also Healthgevity Nation. But the bridge is, if you want to change behavior, you must maybe associate with MIT, with the Future You app. Because when you put down the future you is like imagining, what are the scenarios of your future. If you put in then also the genetic data into that, then of course, when people can see the scenarios of playing out, this is what is going to happen with that genome and that, and that. If you don't change your behavior, I think there is far more chance for people to really do something about their, their behavior, their actions, their decisions, and their choices, if they really start looking at this scenario thinking, which is of course what we need then to put into education, not only in children's education, but also in adult education. It's like future thinking is the future, and it's about better present.
Super, thank you for that. Another question? Yes, please, the lady behind the gentleman who just spoke.
Thank you. So I just want to thank all the panelists. This has been really insightful. So my name is Sarah, and my question is more of like a chicken or egg question. But when you're talking about behavior, you're talking about environment, how do we weigh those? Is it, you have a particular predisposition, you start with like a behavior, and then you kind of influence the environment? Or is the environment kind of influencing the behavior? Because you mentioned smoking, but because of governments actually making it legal, having that access, as soon as they started kind of putting warning labels, we saw a shift in behavior. But how does that link to individuals that might crave nicotine, for example, when they're stressed? So I'd love to just hear your perspective.
Laura, do you want to jump in on that?
Yes. Since the majority of my work has been in the genetics of smoking and understanding this behavior, if smoking doesn't exist, it just doesn't matter what your genetic predisposition is. It's just not an environment that's there. And thinking about smoking really is a 20th century behavior. That's when the tobacco industry developed and smoking became prominent across the world. So what we've seen is government regulation has really, business and government regulation have promoted the behavior and then decreased it. But what we could do now, which is really amazing, is we could identify this small group, 10% of people who smoke, that cannot quit. because most people quit. They understand what their risk is, and people quit generally when they're in their 40s. But there's this small group that cannot quit. So we could now identify them. So environment is critically important.
So that's the equivalent, Laura, in my mind, of what you were saying with a super high-risk group that may be potentially suffering a disease in the future. And so you're basically identifying cohorts that need special attention and designing behavioral programs to support them.
Yes.
Is that
and that's the group that really needs that. So we do have to think of environment all the time with it, and we want to promote a healthy
Gabby, you were going to go on?
Yeah, just very briefly that there is a massive problem with pollution as well, which is the same area of thinking, right? So there is strong evidence that pollution can initiate disease, and we can intervene there as a larger level of...
We're going to take one more question. The gentleman behind the lady who just spoke, if I can follow that line back, we can get
sorry,
a mic.
It's me, yeah. Hi, sorry.
Oh, sorry, we've jumped. That's fine.
That's all right. So I'm Vinod Gaba, based in Dubai. So I'm trying to understand how much you all are leaning towards the startup ecosystem for solutions to these problems. For instance, we're working on 2 solutions. One is using these polygenic risk scores and converting them to actionable insights and then influencing behavioral modification using technology. So that already is in place and in the population, slowly penetrating. The second is actually, highlighting the need for larger data sets, particularly for more rare scenarios, and the fact that you're all sitting on siloed data at the moment, but unable to communicate those because there's insufficient safe and encrypted mechanisms to do that. And we've effectively already developed that for the genetic space. So I'm just wondering, how much are you guys looking out towards the startup ecosystem to solve some of these identified problems? Thank you.
JJ, do you want to comment?
Yeah, PRS, polygenic risk score is a hot topic. My opinion is we are only the starting point, like I explained.
You're right.
At this moment, we feel the polygenic risk score is a fixed effect, but that's wrong. Because we calculate the risk based on how many allele you carry, but not talk about how many allele is active in you. That information we have not there yet. The long way to go. I believe in the future, the polygenic risk score calculation will become dynamic. Based on the current condition, the information come in, will modify the calculation because each allele expression level become different. Then you count real risk, or that time called the real genetic risk at the moment. That will tell people, motivate people to do something, because otherwise I do everything, my genetic risk score is still the same. Why do I do that? But that's wrong, because that genetic risk score is different. You do something in the future, not today, you will see your score changing. That is a strong motivation for people to act.
I will thank you for that, JJ. I'll just comment, if I can, on the startup ecosystem. I think that was the first question you asked. Certainly from my perspective, I've been involved with a number of these programs over the years. Great opportunity for partnering, collaborating across the breadth of what it takes to run these incredibly complex programs, right from how you consent people appropriately, manage samples, store biobanks, look at sequencing, generating outputs across multi-omics. You need partners across the board in this ecosystem. The data play in terms of primary, secondary, tertiary analysis, huge opportunity to create an ecosystem, very, very open to understanding where those collaborations might work. There's lots of overlaps between different companies, but there's a wonderful opportunity to facilitate, accelerate, engage externally with partners around that agenda. Certainly from our perspective, we're very open to that. And your point, which I think is also very pertinent about locked up data sets in different parts of the world, I think we're starting to recognize that, looking at federated approaches, looking at driving collaborations across countries. We do need to get ourselves clear and sorted and make sure the right governance is around the individual data sets before we start collaborating. But there is absolutely no doubt, ultimately, this will be a global endeavor. Do I have time to take more questions? 2 more questions. The gentleman who was in front of the person who asked last time, and then I will jump to the gentleman on the left there.
Thank you very much. Jamie Metzel. Can you please talk a little bit more about the path toward understanding more of complex dynamic systems biology? I mean, we're breaking it down into these little pieces, but the coin of the realm, it seems, will be putting all these systems together. What's the path towards understanding it, and what will be the implications for human healthcare?
Who wants to take that?
I have
JJ.
my sentence, few sentences on that. Right now, we are very good at goal scales, population level. Okay, there are actually two fields. If people know that there is ongoing effort to actually sequencing gene expression every individual cell, because the single cell sequencing project. People talk about how many cells do we need sequencing that individual to be able to have a full understanding about the expression program we talk about. 1,000, not enough. People talk about millions, right? Well, that's where people think the million cell will give you a full presentation of that individual. Same time, we have the UAE program to sequence individual, 1 million people. When will become 1 million times 1 million? Can we sequence 1 million cells from 1 million people? Would that be the data set we require to have a full understanding of what is going on? That probably the future. That's where we move into. We thought we did a very thing big enough. I don't think so. We are only scratch a certain area. There's a long way to go.
I want to add just a little bit to this. So when you think of the polygenic scores that we have, they're looking at risk for diseases. But when you get to the biology and the biomarkers, you really don't need the polygenic scores because you have the biology about what's going on. And so where I think the polygenic scores are important is predicting who's at risk for then getting these biomarkers, which then lead to disease. And we just do not know enough about really the mechanisms of disease. But we have information already that we can give, and we should not wait until we know every single mechanism.
Super, thank you. And then the final question.
Oh, Gab.
Sorry, Gabby, did you?
Yes.
Sorry, apologies.
So,
Gabby, please respond.
My comment would be that we do need dynamic systems. We do need to think about experimentation. That's our most valuable thing. Like in animal models, you can actually take cord blood. That would be my proposal to collect cord blood that probably is discarded. That's a massive resource. And we can analyze the human blood stem cells and its differentiation in animals that have been, say, irradiated, and they will repopulate it with human cells. We can understand the dynamic systems across differentiation stages. So that's somewhere where systems physiology would have place.
We can continue the dialogue. There is one question left. Apologies, we're run out of time, but we can, of course, continue the discussion.
Hello, everyone. I think it was a great discussion. My name is Mukbil. I am a technology journalist from India. My question is, it's a comment also. It's also drawing on other disciplines, and I will be very brief because I know the time is in short supply. My question is related to the fact that on one hand you have the potential to gather huge amounts of data, and on the other I am drawing from industries where you have a concept called a digital twin. Now what is a digital twin? You create a digital version of, say, an automobile, and when something happens, you go back to that and you try and fix that. Do you think in the future, because we are talking about the future, in 100 years from now, would it be possible to have that kind of a data where you can create a digital twin which absolutely relates to that specific human being? I mean, it's a very futuristic concept.
Great question. Quick, short answer, if you would,
Thank you.
each of you, and maybe this will be the closing comment.
Oh, yeah,
Please,
I think it's
Gabby.
a wonderful idea, absolutely. Now, it's a hugely complex system that we have in the body, so if that's possible to be reproduced in silico, absolutely, that would be helpful.
Laura, quick comment?
You know, humanity is incredible, and I love the diversity that we have, and I embrace kind of who I am now, where I am, and going in the future, and I don't know how I feel about that, having my digital twin.
JJ,
Yeah.
final.
Well, I was more optimistic. If we can generate 1 million by 1 million data sets, there is a chance for us to create that digital twin.
Brilliant.
With information there.
Great comment. Nice, very nice finish to the great discussion. Thank you.